Right, so our next speaker is Luis Herrera-Estrella. Known Luis many years, nearly 40 years, and because he's been a real leader in investigating agrobacterium biology and inventing ways of enabling us to use agrobacterium with plant genetic manipulation. And he's gone on to make a number of interesting investigations into plant biology. He's a emeritus professor at Cinvestav, and he's a professor at Texas Tech University. And he's going to tell us about the regulatory network controlling seed desiccation tolerance, that's reactivation in vegetative tissue, because this is a process that plants have to go through between being formed as seed to tolerate that desiccation and then to absorb water and grow subsequently. So Luis, looking forward to hearing your talk.
So thank you very much for the kind introduction and the invitation to participate at this very interesting seminar. And so I'm going to talk about a very fascinating, oops, I cannot, something happened. A very fascinating process that is desiccation tolerance. So desiccation is the capacity of an organism to survive in the dry state for long periods of time. So we have, for instance, the example of Artemia salina, which eggs are viable for over 10 years in the desiccated state, and the little embryos that are in the eggs survive the dry state during this time. And there is also these little animals, the tardigrades or water bears, that are extremely resilient to environmental stress. They can survive 50 years in the desiccated state. And when you water them again, they come back to life. These little animals are also very resilient to radiation. They have been taken to the space station and exposed to cosmic rays, and they can survive very well this very harsh environment. In plants, desiccation tolerance has been a very important process. I had a video here which I cannot play anymore. I don't know why. Oops. So early land plants had to acquire desiccation tolerance to be able to survive. Once they started to move away from the edge of the water, they had to tolerate longer and longer periods of dryness. So they had to evolve mechanisms to survive in the dry state. This is probably inherited from the algae from which early land plants evolved from. So this desiccation tolerance evolved, but perhaps the most amazing process of desiccation tolerance is desiccation tolerance in seeds. So here I have a few examples. So these are, for instance, acacia seeds that were rescued from an herbarium in Iran by a Swedish scientist. And when you take these seeds that are 150 years old and you put them in media, they still are viable and germinate. There are also some other even more striking examples. Lotus seeds from a 1300-year-old thumb in China, in which these lotus seeds were again still viable. And the record so far is 2,000 years old date seeds that were rescued from an Egyptian tomb. And when you saw them in a proper soil with water, they are still viable. So that means that during seed development, plants have to activate a very complex system to protect all the cellular structures, membrane structures, ribosome, DNA and RNA integrity, chloroplast membranes, so that they can survive in the dry state for all these periods of time. So when plants evolve, early land plants didn't have proper root systems and they didn't have vascular systems. So they had to develop desiccation tolerance in the vegetative tissue so that they could survive to the periods of dryness. Later on, plants developed through roots, vascular systems, and they no longer require the capacity to be vegetative tolerant to desiccation. Vegetative tolerant desiccation that was very important in the early branches of plants was lost when tracheophytes evolved. So when the vascular system evolved, the plant had the way to extract water more efficiently from the soil, so they didn't require anymore to have vegetative desiccation. So vegetative desiccation was lost. And it has revolved many times during the evolution of land plants, particularly in the angiosperms, in which a vegetative desiccation has re-evolved at least 13 times. So if we have a system in which vegetative desiccation is present in the early land plants, then it's lost and hijacked to be rewired to be expressed in seeds. And this vegetative tolerance in seeds is then rewired again to be back in the vegetative tissues, suggests that although this is a very complex process that requires expression of late embryogenesis, abundant proteins, heat shock proteins, elite proteins, a mechanism to protect against oxidative stress, reactive oxygen species. So how can all these very complex mechanisms be activated and rewired to be activated in different tissues? So that's one of the questions that we would like to understand and test whether we can actually reconstruct an event of plant evolution. If you have desiccation tolerance in the seed, can we reactivate that pathway in vegetative tissues? So during the process of seed development, the early steps in seed development is mainly cell division and cell differentiation, in which the embryo proper is developed. Then, after a time when you have an almost fully developed embryo, the seed starts to lose water and it starts to accumulate storage compounds, lipids and carbohydrates. At some point during this process, when water is lost, the program to survive in the desiccated state is activated. So all these genes that are required to protect the cell structures to maintain viability is activated. So how can we understand this process? So fortunately, many people for a long time started studying embryo-lethal mutations. The idea was to identify genes that were required for embryo formation and embryo viability. Some of these mutants were actually not embryo-lethal, but they were desiccation intolerant. So if you could rescue the seed before it dried out, the embryo was viable. If the seed desiccated, then the embryo was dead. So these mutants are very interesting because they are mutations in master transcription factor regulators that regulate the transition from the embryo development to vegetative development. So in these mutants, the embryo doesn't go to the desiccation state and dormancy, but they rather directly germinate after embryo formation. And this is seen because you get trichomes in the cotyledons and they remain green rather than becoming white. So there are mutants in these transcription factors which are desiccation sensitive and desiccation tolerant. So they have a very similar phenotype in the transition from embryo to vegetative state, but some lose desiccation tolerance and others don't. So if we compare these mutants, then we can start identifying the genes and the program for desiccation tolerance. For instance, the LEG1 mutant and the LEG2 mutants are very similar. They both accumulate chlorophyll in the dry seed. They accumulate anthocyanins in the cotyledon. They have reduced storage compounds. They produce ectopic trichomes, but one is desiccation tolerant and the other is desiccation intolerant. So if we compare the global transcription in these two seeds during seed development, we should be able to evaluate identify all the genes that are activated to acquire desiccation tolerance. There are other mutants, for instance, AB35 is a mutant which is no longer desiccation tolerant, but there are alleles of AB31 which are still desiccation tolerant, although they have still reduced ABA sensitivity. So we made comparisons of these mutants at 3 different developmental stages during seed formation. So we analyzed RNA-seq data and we analyzed the metabolomic profile of these seeds during the development. So the idea was to see whether we could identify the set of genes that are required for acquiring desiccation tolerance. So we found 2 major sets of genes. Those that are activated in these mutants that are intolerant to desiccation. These genes have to go these genes have to do with the transition from embryonic to vegetative growth because they encode genes involving chlorophyll biosynthesis, antenna formation. And there is 2 other sets of genes which are not activated in the intolerant mutants, but they are activated in the tolerant mutants. These sets of genes are the ones that we believe are responsible for the mechanism. So in the genes that are upregulated in the intolerant mutants, of course, we have photosynthesis, sucrosynthesis, cell division, and cell expansion because these embryos go to the vegetative growth. The genes that are not activated in the intolerant mutants have to do with lipid biosynthesis, storage proteins, and straight related responses, LIA proteins, oligosaccharide biosynthesis, heat shock proteins, antioxidant mechanisms, and so on. So to understand how this is activated, we build a regulatory co-expression network of seed development. So we use about 350 sets of gene expression of seeds in different processes, different mutants. So we build this regulatory network. And in this network, we identify all the genes that are activated, all the transcription factors that are activated during seed formation. And among these transcription factors, we identify those that fail to activate in the mutants that are no longer desiccation tolerant. So in the expectation that these transcription factors are the ones that orchestrate the activation of the desiccation tolerant mechanism. So we found 2 subnetworks that are active, that are not activated in tolerant mutants, but are fully activated in the tolerant mutants. These 2 subnetworks have different properties. One of them has a dual role in activating the expression of genes involved in the accumulation of reserve compounds, and also in genes that have to do with the stress tolerance, for instance, the accumulation of LIA proteins. So we can see here in this network, for instance, all these yellow dots are LIA proteins. Some these other proteins are heat shock. Some of them have to do with the accumulation of sucrose and oligosaccharides that are required for desiccation tolerance. The second subnetwork is fully involved in activating cell protection mechanisms. Again, it regulates the accumulation of many LIA proteins. It also accumulates proteins that are involved in stress-related processes, synthesis of oligosaccharides, heat shock proteins, and so on. These 2 subnetworks change during the evolution of seed development. So the subnetwork alters its architecture during seed development. First, initially, is only involved in the accumulation of reserve compounds. then it starts to activate genes involved in stress tolerance until more or less half of the network is part of stress tolerance and the other half is involved in reserve compounds. Here it's important to mention that at 17 days is when the accumulation of sugars and other compounds are activated during the desiccation tolerance mechanism. The second network is only activated when water is reduced enough in the seed to activate the desiccation process. And this subnetwork also evolves and activates many genes involved in desiccation tolerance. So if these networks are real networks, mutations in the transcription factors that control these subnetworks should lose seed viability during desiccation. So we produce mutants in different transcription factors of these subnetworks. We produce seeds and we evaluated the seed viability after 30 days of desiccation. So as you can see, the wild type has almost 100% of germination, and the different mutants have different rates of survival after desiccation, suggesting that in these transcription factors have to do with desiccation tolerance. If it is a network, double and triple mutants should have further reduced their tolerance to desiccation. And that's the case if we have a double mutant or a triple mutant, then the seed becomes almost desiccation intolerant. To confirm that these transcription factors had to do with desiccation tolerance, we ectopically express some of these transcription factors in the AB35 genetic background. AB35 is completely desiccation intolerant. And we can see that the ectopic expression of some of these transcription factors partially rescue the capacity of the seed to survive short periods of desiccation, suggesting that indeed these networks exist and these nodes, which are transcription factors, are indeed coordinating the expression of a large number of genes involved in desiccation tolerance. So what we want to do is to reconstruct this event in vitro. What happens if we are reactivate in vegetative tissues the expression of these networks? So to assay for this desiccation tolerance, what we do is we germinate the seeds for 14 days. After 40 days, we have 10 days of full irrigation, and then we stop watering for different periods of time. It could be 2 weeks, 3 weeks, 4 weeks. And then we do a recovery irrigation treatment, and we count the plants that actually survive the period of dryness and how many of them die. So we have done these experiments using the expression of different combinations of 2 or 3 different transcription factors. And you can see that the survival of the plants to 30 days of desiccation increases when we express several genes. And when we reach a whole one of the subnetworks, when we express the 5 most important nodes of subnetwork 2, we almost reach 100% of survival after 30 days of desiccation. This is the plants, how they behave. This is the control in which we stop watering. We did the recovery irrigation, and the plant, all of them die. These are expressing 5 transcription factors. We desiccate them, we water, we do a water recovery, and they all survive. Here it is interesting to note that if you constitutively express these transcription factors, they reduce the growth of the plant because probably constitutively activating the expression of LIA proteins, heat shock proteins, the accumulation of oligosaccharides required for desiccation tolerance has a big penalty in growth. If we induce these genes by an inducible, drought-inducible system using promoters that are responsive to drought, then we can see that the plants grow normal, but they are still fully resistant to this process of desiccation. These plants are still not yet 100% desiccation tolerance. 6 months without water, they die. So, but we are now trying to activate the 2 subnetworks using the CRISPR-Cas system for gene activation to see if we can orchestrate the activation of 2 sets of subnetworks and see whether we can actually reconstruct this evolutive event in nature. Of course, activating these genes also provides some information about how to design plants that are drought tolerant. So depending on the combinations of transcription factors we activate, we get different levels and different mechanisms of drought tolerance that might be useful for the future breeding of drought tolerant plants. And this is just my research group, and the people working on this topic is Sandra Gonzalez, Telma Rico, and Gerardo Alejo. And I thank you for your attention. I'm sorry that my video didn't run properly. I don't know what happened. Thank you very much.